Hello AP Bio students, and welcome to our video on facilitated diffusion. Now, facilitated diffusion is basically diffusion with help, and it's a lot of like riding your bike to school and having a hill. Let's talk about what we already know. We know that in diffusion, molecules move from an area of high concentration to an area of low concentration until they've reached equilibrium. Now, I think a lot of this reminds me of the horrid smell that you smell about 3 blocks away from Abercrombie and Fitch in San Luis. So what happens there is there's this person, and they are paid to routinely go around and spray the store with their horrid smelling perfume. So they go around and they're like, squirt, 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 spray, 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 right? Oh, it smells so good. Well, now we have this high concentration of perfume within the store. So where does it go? Well, they happen to always keep their doors open. So it's going to move from area of high concentration to an area of low concentration, which could be like 3 blocks away. I call it smell pollution. So the way I think about diffusion also is that it's like a bike. And it's like you're riding your bike to school, and you're so lucky as to get a downhill. So you're pedaling and pedaling, and all of a sudden you get this break. You're moving in the direction you want to go. However, you have to give no energy to go that direction. So diffusion. Let's look at facilitated diffusion. Now, facilitated diffusion does not use ATP. We know that ATP, adenosine triphosphate, is the molecule used to do work in our cells, and facilitated diffusion does not use this. So what does it use? Well, it uses our superstars, proteins, and it uses a bunch of different type of proteins. Let's talk about them. The first type of protein I'd like to talk about is a channel protein. Now, a channel protein has a specialized corridor which uniquely matches the molecule that it wants to move into the cell or out of the cell, and it basically speeds up diffusion. Now, one thing to note is that this is moving in the same direction as the concentration gradient. So remember, the concentration gradient is just the direction of diffusion. So we would have high concentration of solutes here, low concentration of solutes here. Now, why would this be needed? Well, a lot of times these solutes are too large to actually get through the membrane. So what happens, remember the membrane is selectively permeable. So what happens is these things need to be moved in and out of the cell. So we have these wonderful channel proteins that allow that to happen. I think of it, and this analogy is a bit of a stretch. Let's say we all decided that, you know what, life's just short. Let's just eat ice cream all day and not do anything. So, a few years later, we're all sitting around and we're 500 pounds. And we're like, well, I guess I put on a little weight, but oh well. We're big mamas. We're like these large solutes that need to get around, right? So what happens is, let's say we're at a subway station and we have about 30 minutes till our next train comes. Well, the normal seats are a little too teeny for us. We can't quite squeeze through. Luckily, we have these large seats that allow us to fit right in. It's a lot like a channel protein. They are large enough for these large macromolecules to move in. So I think of facilitated diffusion in this sense as your parents saying, you know what, you're doing so good, you're taking AP biology, you work, we're going to upgrade your bike to a motorcycle. So now you get to ride to school in a motorcycle. However, the motorcycle does not have a working engine. So it's a little bit of a rough going up hills, right? However, once you go downhill, you know about mass and inertia, that a motorcycle will actually end up going faster downhill because it has more mass. So it's a lot like diffusion. We're still going in the direction we want to go with the concentration gradient. However, we're able to go much faster. Okay? So let's talk about some examples of these channel proteins. One example, a superstar, is an aquaporin. And an aquaporin is a channel protein that's main job is to move water, and to move it fast and to move it well. And this is really important in plant cells and kidney cells and red blood cells. Have you ever seen a plant, and maybe you have a house plant, and you see it and it's all wilty and sad, and you're like, oh no, I need to water it. So you douse it with water, and you're like, I hope it's not too late. So you come back a half hour later and the plant's all perky, like nothing ever happened, right? So that plant had specialized aquaporins in its cell to move that water really fast, to change those plant cells from being flaccid to turgid, so we have nice perky leaves, so that photosynthesis can happen very efficiently. Another reason to be thankful for these channel proteins is because there's a lot of these aquaporins within your kidney. And we know that your kidney is kind of the filtration monitor of our blood and all the toxins and everything that accumulate, and is in charge of filtering it and getting rid of the waste. So our kidney cells have a ton of aquaporin, and their whole job is to reclaim water from urine before you pee. Now, if your aquaporins did not do this, you'd have to drink 50 gallons of water per day and pee 50 gallons of water per day if the kidneys did not perform this function. So thank goodness for aquaporin. I think there should be like a aquaporin day, like a holiday, right? Let's move on. Our next type that I like to talk about is, it's a channel protein as well, but it's gated. And a lot of times we call these gated ion channels. Ion channels. And what happens is it's a 2 protein complex, and we have our first protein is a channel protein, but it's got these gates that kind of keep things from flowing in. And it has this associated protein, which is the receptor protein. It's got this nice little receptor molecule, which is probably a carbohydrate, that sits around and waits for a ligand. Now this ligand, once engaged the receptor, signals to the channel protein saying, hey, you can open the gates, this guy's cool, he can come in. So I think of it, a lot of it like St. Peter, who waits around at the pearly gates and decides who goes in and who doesn't based on the signal from God. So those are our gated ion channels. The last one I'd like to talk about is a super awesome, crazy cool protein that we actually still don't totally understand. It's a carrier protein. Now you may think that a carrier protein uses ATP, but it does not. It's a superstar. So what happens is a carrier protein, the solute that it needs to transport actually binds to this kind of active site. And what happens is that triggers a change in shape to move the active site so that the solute can freely move into the cell. Now, once again, this is going with the concentration gradient. And it's a really cool process. No energy is used, and the solute actually attaches to the active site. The protein changes shape and is able to move the solute into the cell membrane. A great analogy of this is the Panama Canal. If you haven't seen a video of the Panama Canal working, it's absolutely amazing. So what happens is we have this boat, and this boat would symbolize the solute, right? And the solute needs to go from the Pacific Ocean to the Atlantic Ocean. And what needs to happen for it to get through is it moves into this kind of gated area. And what happens is the gate behind it closes, and then that area fills up with water, and the boat actually becomes elevated, right? Because the water level goes up. So it becomes elevated enough so that it can move up to the next level, and again and again and again. So essentially, this canal is changing shape to move this boat through. So that's a lot like our carrier proteins. Now, things can go really wrong when our carrier proteins don't work. There's a mutation that causes a disease called Cystinuria. Cystinuria. And this is a genetic disorder, basically a mutation in the DNA that causes specific transport proteins that move the amino acid called cysteine out of your kidneys and back into your bloodstream. So usually these transport proteins or carrier proteins are involved in getting this amino acid and kind of recycling it and returning it back to the bloodstream. Well, when your transport protein doesn't work because there was a mutation, so it's a weird shape, the cysteine doesn't fit in it, it can't function correctly, these transport proteins are ineffective. So you get an accumulation of cysteine within the kidneys. And that's a bummer because when cysteine accumulates and hangs out for a while, it actually forms like a crystalline stone. And you get what's called cystine stones. And if you've ever talked to anybody who's had a kidney stone, they'll tell you it's one of the most painful things they've ever experienced. So this is not a genetic disorder that you want to have. In review, we talked about osmosis and diffusion, which was also a review, moving from high concentration to low concentration. And we talked about how facilitated diffusion basically makes this whole process move a lot faster, and it does it without using ATP. We talked about a few different proteins that help that process. We have the channel protein, which is like that corridor that allows specific molecules to go through at a faster rate than diffusion. And our example of that is aquaporins in plants and kidneys. We also had gated ion channels that wait for a ligand in order for those gates to open and allow those solutes to move through. And we had carrier proteins, which actually change their shape to move solutes from one side of the cell membrane to the other. And that concludes our video on facilitated diffusion.